The cerebellum receives vast amounts of information from the spinal cord, brainstem, and the cerebrum, integrates that information, and then transmits the integrated information back to the cerebrum, brainstem, spinal cord, and other areas. In this Cerebellum Advanced Part 2, we'll learn about the input pathways that allow the cerebellum to receive a vast amount of information. In this Cerebellum Advanced Part 2, we'll first outline the cerebellar input pathways, then discuss specific pathways, describing the characteristics of the posterior and anterior spinocerebellar tracts, followed by the reticulocerebellar tract and the largest input pathway, the cerebro-ponto-cerebellar tract, before finally explaining the olivocerebellar climbing fiber connection. This table summarizes the primary cerebellar input pathways, detailing the neural tracts, types of nerve fibers, origins, cerebellar peduncles passing through, terminations, and whether or not decussation occurs. As explained in the introductory section, input fibers to the cerebellar cortex consist of two types: climbing fibers and mossy fibers. The olivocerebellar tract is the only pathway composed of climbing fibers, while all other input pathways consist of mossy fibers. Regarding the cerebellar peduncles that tracts pass through, only the pontocerebellar tract passes through the middle cerebellar peduncle, while the anterior spinocerebellar tract crosses twice and passes through the superior cerebellar peduncle. All other input pathways pass through the inferior cerebellar peduncle. Although the olivocerebellar and pontocerebellar tracts decussate, the majority of input pathways do not decussate, resulting in the ipsilateral cerebellar function. The details of these major input pathways are described below. We'll explain the spinocerebellar tracts as the initial input pathways to the cerebellum. There are multiple input pathways from the spinal cord to the cerebellum. Some pathways, most notably the posterior spinocerebellar tract, connect the spinal cord directly to the cerebellum without interruption, and are thus termed direct spinocerebellar tracts. In contrast, other pathways, such as the spino-olivocerebellar tract, involve synaptic connections within brainstem nuclei and are therefore referred to as indirect spinocerebellar tracts. Functionally, the direct spinocerebellar tract conveys two types of information. One type consists of information from muscle spindles, tendon organs, and low-threshold cutaneous mechanoreceptors. This allows for the rapid transmission of precise temporal information regarding movements and cutaneous stimuli to the cerebellum. The other type of information does not originate from sensory receptors, but instead conveys information regarding spinal interneurons that mediate spinal reflex arcs and similar circuits. The spinocerebellar tracts originate from neurons in the spinal cord gray matter, such as the posterior thoracic nucleus, also known as Clark's column. The fibers of these tracts ascend within the lateral funiculus of the spinal cord. Their course follows a somatotopic arrangement, ensuring that information from different body regions remains segregated. The somatotopic arrangement observed in the spinocerebellar tract is preserved in the cerebellar cortex. Within the anterior lobe, the lower limbs are located anteriorly, while the upper limbs and face are positioned progressively more posteriorly. In contrast, the arrangement in the posterior lobe is reversed, with the face located anteriorly. We'll explain the posterior spinocerebellar tract and the anterior spinocerebellar tract as representative examples of direct spinocerebellar tracts. The posterior spinocerebellar tract is also called the dorsal spinocerebellar tract. The posterior spinocerebellar tract originates from the posterior thoracic nucleus, also known as Clark's column, located in the lateral column of the dorsal horn within the thoracic and upper lumbar spinal cord, spanning segments T1 to L3. This nucleus receives all types of primary afferent fibers, including those carrying information from lower limb muscles and joints, as well as cutaneous sensory data, via the gracile fasciculus. Consequently, the posterior spinocerebellar tract transmits proprioceptive and exteroceptive sensory information from the ipsilateral lower limb to the cerebellum. The axons of this tract, measuring 20 micrometers in external diameter and the largest in the central nervous system, enable the rapid transmission of movement-related information. The posterior spinocerebellar tract enters the inferior cerebellar peduncle without decussating, gives off collaterals to the intracerebellar nuclei, and finally terminates in the cerebellar cortex of the lower limb region. Here is a summary of the posterior spinocerebellar tract. It originates in the posterior thoracic nucleus, does not decussate, and transmits proprioceptive and exteroceptive sensory information from the ipsilateral lower limb to the spinocerebellar cortex. We'll explain the cuneo-cerebellar tract. The upper limb equivalent of the posterior spinocerebellar tract is the cuneo-cerebellar tract, which contains exteroceptive and proprioceptive components, which originate from the accessory cuneate nucleus in the medulla oblongata. The tract enters the cerebellum via the ipsilateral inferior cerebellar peduncle without crossing. Next, we'll explain the anterior spinocerebellar tract, also called the ventral spinocerebellar tract. The anterior spinocerebellar tract runs within the lateral funiculus of the spinal cord, immediately ventral to the before-mentioned posterior spinocerebellar tract. Its cells of origin are located in the intermediate gray matter, rexid laminae 5 to 7 of the lumbosacral spinal cord, and they transmit spinal reflex activity related to the lower limbs and lower trunk to the cerebellum. The anterior spinocerebellar tract decussates to the contralateral side near its origin, ascends near the surface of the spinal cord as far as the lower midbrain, and then turns inferiorly to travel within the superior cerebellar peduncle. Subsequently, the majority of the fibers decussate again to the contralateral side within the cerebellum and project to the anterior cerebellar vermis. Their distribution within the intracerebellar nuclei and the cerebellar cortex is similar to that of the dorsal spinocerebellar tract. Although both the posterior and anterior spinocerebellar tracts convey proprioceptive and exteroceptive sensory information from the lower limbs, they differ functionally. Neurons in the posterior thoracic nucleus, the cells of origin for the posterior tract, transmit information characterized by modality and spatial specificity, derived from 1A, 1B, and group 2 muscle afferents, thereby facilitating fine, coordinated movement of individual limb muscles. In contrast, neurons of the anterior spinocerebellar tract convey information from extensive sensory receptor fields. Without functional subdivision, they contribute to the synergistic movement of the lower limb as a whole and to postural control. Next, we'll explain the reticulocerebellar connection. There are three nuclei that serve as the origins of the reticulocerebellar tract, projecting from the brainstem reticular formation to the cerebellum. The nucleus reticularis tegmenti pontis, NRTP, and the lateral reticular nucleus, LRN, and paramedian reticular nucleus, PRN, of the medulla. Each of these reticular nuclei receives inputs from sources such as the cerebral cortex and spinal cord. Their output fibers travel by the inferior cerebellar peduncle and project ipsilaterally or bilaterally to the cerebellar vermis or the cerebellar cortex. Next, we'll explain the cerebro-ponto-cerebellar pathway, a system of afferent fibers projecting from the cerebral cortex to the cerebellum. The largest group of afferent fibers in humans originate in the pontine nuclei. These fibers travel via the middle cerebellar peduncle as the pontocerebellar tract and terminate primarily in the cerebellar hemispheres. Since the pontine nuclei receive input from the cerebral cortex, this system constitutes the cerebro-ponto-cerebellar pathway. The largest input to the pontine nuclei originates in the cerebral cortex, forming the cortico-pontine tract. The pontine nuclei process information received from the cerebral cortex and transmitted to the cerebellar cortex via the pontocerebellar tract. While the cortico-pontine tract is uncrossed, the majority of pontocerebellar fibers cross the midline. Consequently, the cerebral cortex primarily influences the contralateral cerebellar hemisphere. The fibers constituting the cortico-pontine tract originate primarily from the primary motor cortex, MI, and primary somatosensory cortex, SI, but they also arise from the supplementary motor area, SMA, the premotor area, PMA, and areas 5 and 7 of the posterior parietal lobe. The cerebellum modulates the activity of these motor areas to ensure that movements are executed smoothly and accurately. Corticopontine fibers are arranged according to functional topography, with distinct cortical regions connecting to specific sub-regions of the pontine nuclei. Specifically, fibers from the primary motor cortex and the primary somatosensory cortex project to separate areas within the pontine nuclei. The corticopontine tract passes through the internal capsule to reach the midbrain, where it travels within the cerebral peduncle. Although the human cerebral peduncle contains approximately 19 million nerve fibers, the so-called pyramidal tract accounts for only 1 million of them, indicating that corticopontine fibers constitute the majority. Within the cerebral peduncle, these fibers exhibit a functional arrangement. Fibers from the frontal lobe occupy the medial aspect, while those from the parietal, occipital, and temporal lobes occupy the lateral aspect. The neural pathway extending from the cerebral cortex to the pontine nuclei exhibits marked divergence. For instance, a small region of the primary motor cortex projects to numerous neuronal groups within the pontine nuclei. Conversely, individual neurons in the pontine nuclei receive convergent inputs from multiple cortical neuronal groups, integrate this information, and output the integrated signal to the cerebellum. The corticopontine cerebellar pathway integrates a vast array of inputs from the cerebral cortex and projects them to the cerebellar cortex. Next, will explain the olivocerebellar climbing fiber connections. The inferior olivocerebellar complex is subdivided into three nuclei. One is the largest principal or inferior olivocerebellar nucleus, which presents a complex, convoluted structure. Second is the smaller dorsal accessory olivocerebellar nucleus, and the third is the smaller medial accessory olivocerebellar nucleus. Nerve fibers originating from the olivocerebellar nucleus form the olivocerebellar tract and reach the contralateral cerebellar cortex, while ascending collateral branches to the lateral vestibular nucleus and the intracerebellar nuclei. Olivocerebellar fibers become climbing fibers and terminate on Purkinje cells arranged in longitudinal rows perpendicular to the cerebellar medulla. A prominent feature of the olivocerebellar pathway is the zonal organization observed in the termination areas of climbing fibers. Cerebellar cortex is divided into anterior-posterior or sagittal zones, designated A, B, C1, C2, C3, and D from medial to lateral, with each zone receiving projections from specific regions of the contralateral olivocerebellar nucleus. For instance, the A zone receives projections from the caudal part of the medial accessory olivocerebellar nucleus, while the B zone receives them from the caudal part of the dorsal accessory olivocerebellar nucleus. The principal olivocerebellar nucleus projects to the contralateral cerebellar hemisphere, D zone, demonstrating a strict topographical arrangement between the inferior olivocerebellar complex and the cerebellum. As before mentioned, this zonal organization within the olivocerebellar pathway also applies to the projections from the cerebellar cortex to the intracerebellar nuclei. The inferior olivocerebellar complex receives both ascending and descending inputs. The ascending input fibers are predominantly crossed. Traveling by the posterior columns as spinocerebellar fibers, they project mainly to the medial and dorsal accessory olivocerebellar nuclei. Descending inputs are uncrossed. Fibers originating from the ipsilateral cerebral cortex, thalamus, red nucleus, and the periaqueductal gray matter of the midbrain. In particular, input fibers from the red nucleus and the periaqueductal gray matter of the midbrain become the central tegmental tract and form the alveoli amygdalum. In this Cerebellum Advanced Part 2, we covered an overview of the input pathways that allow the cerebellum to receive vast amounts of information, as well as the characteristics of specific pathways. That is, the spinocerebellar, reticulocerebellar, cerebro-ponto-cerebellar, and olivocerebellar climbing fiber tracts. The Cerebellum Advanced series continues with Part 3.